Hello students. So today the topic is transcription. Okay. So what is transcription? The process of copying genetic information from one strand of DNA into RNA is known as transcription. Or in simplify the words, we can say that the formation of RNA from DNA is called transcription. So this is also called as heterocatalysis. Heterocatalysis or transcription. Already we discussed under functions of DNA, the two major functions of DNA. One is replication and the other one is transcription. Replication occurs only at the time of cell division, that is in eukaryotes. And in prokaryotes, of course, it occurs at the time of binary fission. Okay. But whereas transcription, that is RNA formation, occurs all the time, every time, wherever there is requirement in the cell. But what is the difference between replication and transcription? During replication, DNA will double. The DNA content of the cell will double. And whereas in transcription, and you just say, it is copying genetic information present on DNA in the form of RNA. And here in the definition, your focus should be on this one, that is from one strand of DNA. So remember one thing, that during transcription, suppose this is DNA, and only a segment of DNA will undergo transcription. This is the segment. And now among the segment can be called as transcription unit. Transcription unit. And remember also that the segment itself is nothing but what we call, that is the gene. But the problem here is this gene or the segment is having information for mRNA, that is messenger RNA, and it has information for ribosomal RNA, that is rRNA, and it has information even for tRNA, that is transfer RNA. So then it becomes complicated, because we know that the gene carries the information for a particular character. So the hereditary unit is called what? Gene. So then this mRNA actually carries that information, and it will undergo translation and produces what? A protein. And that protein, when it behaves as enzyme or when it is a proteinaceous enzyme, so it catalyzes the reaction, and as a result of which the product is formed. And suppose the character is growth, so then the product will be a growth hormone, and that promotes growth in that organism. So that's why now the problem here is the same segment, that is a segment, is having information for mRNA as well as rRNA, as well as tRNA. That's why a scientist called Benzer, he has used one term for this. That is what cistron. Okay, and what is the cistron? The cistron is the unit of DNA or segment of DNA having information for the formation of a polypeptide. Polypeptide. I hope you're understanding what is cistron. Unit of DNA or segment of DNA having information for the formation of a polypeptide or protein. So, like that you have to say this one. And one more thing. Now, let us see. Out of the two strands, if both strands are having the information for the formation of this RNA, then it becomes what? A complicated thing. Means what? The gene expression will become complicated. Because I hope you have to understand this point. A gene having information for the expression of a character, okay, will be expressed in the form of mRNA, okay, and this mRNA will undergo translation, as a result of which a proteinaceous enzyme, a protein or the enzyme is formed, and that enzyme catalyzes the reaction and a product is formed. So I hope you understand it. So this segment of DNA is having information for a particular trait or the character, and we are calling it as what? Gene, and that gene is transcribed in the form of mRNA, and as a result of which this mRNA underwent translation and produced a protein or enzyme, and that enzyme catalyzed the reaction and helped in the formation of the product. Okay, but the thing is that this segment is having two strands, and out of the two strands, only one strand is transcribed into RNA, not both strands. Why both strands are not transcribed? If both strands are transcribed, and you know that these two strands are complementary to each other, and the sequence is not same, it is different. Because of which, if both strands, okay, if both strands are transcribed, so then what will happen? So they produce two different types of mRNAs. Two different types of mRNAs. And these two different types of mRNAs will produce two different types of proteins. And these two different types of proteins will produce two different types of products. Then the gene expression will become complicated. I hope you are understanding. So that's why remember one thing, that out of the two strands, only one strand is behaving as, okay, template strand. Only one strand will behave as template strand. So this is quite opposite to the replication. So in replication, during replication, both strands of DNA will behave as template strands. And one more difference, that during replication, the entire DNA, okay, the entire DNA is replicated or duplicated in the organism. Means what? Both strands are behaving as templates, and the entire DNA of the organism is replicated, as a result of which two daughter DNAs are formed. That is during replication. But what about this transcription? Only one strand is behaving as template strand. And you have to understand one more point here. and which strand will be that. And you know that even this transcription is catalyzed by the enzyme RNA polymerase. RNA polymerase enzyme. And this RNA polymerase enzyme also adds the nucleotides according to the principle of complementarity. According to the principle of complementarity. Means what? If A is there on the strand, it will bring RNA polymerase will bring. Okay, it is not DNA polymerase, that's why it will bring uracil. And if it is T is here, and it will bring adenine. Okay, adenine nucleotide. And if there is C, G. If there is G, C. Like that. So it will be. But don't forget that just like the DNA polymerase enzyme, so the polymerization direction will be from 5' and to 3' direction. So that means this template strand should have what polarity? The template strand polarity should be 3' to 5' polarity. So this is template strand polarity. And this is RNA. Okay. Now let us say generally people think that once one strand will behave as template strand and the next time the next second strand will behave as template strand. It is not the correct idea. Okay, what happens? So only one will behave as the template strand always. And which one will behave as the template strand? The one with polarity 3' to 5' polarity. So this will behave as the template strand. So why only one is behaving as a template? That already I told you. Because only one protein should be formed and one product should be formed. Is what it should have information for only one gene. Okay, that's why that is the thing. And one more thing, suppose why both strands are not behaving as template strands. One more explanation. Let us see the second explanation. Suppose imagine that this strand and these strands, these two strands are unbinded and they got separated, and both are behaving as template strands. Let us imagine both are behaving as template strands, and both are transcribed simultaneously. And if both are transcribed simultaneously, one mRNA is found here and one mRNA is found on this template. Then these two mRNAs are complementary to each other. The two strands are, the two mRNAs are complementary, and because of which, again, there will be formation of hydrogen bonds. As a result of which, double-stranded mRNA is formed. Okay, actually, this is not happening. Just we are okay, discussing what happens, what will be the consequence if both strands will are transcribed simultaneously. So double-stranded mRNA is not at all useful for translation. It cannot be translated because at the time of translation, so okay, so the tRNA will decode the information present on mRNA. So for that purpose, this mRNA should be okay single-stranded. So these are the two explanations why both strands are not behaving as template strands. Okay, we will discuss about that okay transcriptional unit. Okay, now let us say the segment of DNA which is participating in transcription is called transcription unit. What is this transcription unit? The segment of DNA which is participating in transcription is called transcription unit. So this transcription unit is having three components. So the three components of transcription unit are a promoter, two, structural gene, and a terminator. Let us take this diagrammatic representation. These two are the two strands of DNA, and we know that they're anti-parallel, and one is showing 5 prime 3 prime polarity, and the other one is showing 5 prime 3 prime polarity in opposite direction. Okay, as we have already discussed that this polymerization direction is 5 prime to 3 prime. So that means only one will behave as the template strand, and here I have used two different colors for differentiation. The red colored line represents what strand? Template strand. Template strand. And this is also called as non-coding strand or anti-sense strand. You have to memorize these alternate terms. So the template strand is also called what? Non-coding strand or anti-sense strand. And the polarity of this template strand is 3 prime to 5 prime. 3 prime to 5 prime polarity. And now the opposite strand, which is not transcribed, is called non-template strand. I have used black color for this non-template strand. And it is also called as coding strand because it is identical to the mRNA that is formed. Means what the sequence that is present on mRNA is almost similar to the sequence present on coding strand, except that in the place of thymine, you will find uracil in RNA, whereas here you will find thymine. That's why it is called coding strand or sense strand. Okay, in this transcription unit, so a sequence, a specific sequence, which is recognized by RNA polymerase enzyme, is called promoter region or promoter site. And at this end, you will find what? A terminator sequence, and where this transcription will be stopped. Okay, and between this promoter and terminator region, the actual information for the formation of mRNA is present here, and that segment or this part is called what? Structural gene. This is called structural gene. So I hope you are understanding. The structural gene is present between promoter and terminator. Okay. Right. Now, how this promoter, how this transcription occurs, that we will see. But remember one thing, the promoter region is present towards 5 prime end. Means what? Okay. And terminator region is present towards 3 prime end. And you may get confused again. But no confusion. That means it is done with reference to the opposite strand, that is non-template strand. You can see that promoter region is this actually. So this is present opposite to this non-template strand of which polarity? 5' polarity. And the terminator region, okay, is present opposite to 3' polarity. So that's what you have to remember. The promoter region is towards 5' end of a non-template strand, and terminator region is present towards 3' end of this non-template strand. Okay, and how this transcription occurs, that we will discuss in detail later. But you remember also that the structural gene is called as cistron. This structural gene can be called as cistron here. And the reason that this cistron, if it is having information for one polypeptide gene, the cistron having information for one polypeptide gene. Okay. Then the mRNA, when it is formed, okay, will have information for one polypeptide chain, and that's why it is called monocistronic mRNA. Monocistronic mRNA. I hope you're understanding what is this monocistronic mRNA. mRNA carrying information for one protein or one polypeptide is called monocistronic mRNA. This is common in eukaryotes, but in prokaryotes, this cistron will have information for more than one polypeptide. So when this cistron is having information for more than one polypeptide, then when the mRNA is formed, the mRNA will carry information for more than one polypeptide. That's why that mRNA is called polycistronic mRNA. Polycistronic mRNA. Okay, I hope you're understanding. We can recognize two types of mRNAs based on that information. If the mRNA carrying information for one protein is called monocistronic mRNA, common in eukaryotes, and the mRNA which is having information for more than one protein is called polycistronic mRNA.